CRISPR gene editing, illustrated and voiced by Govindi Ramchand and Sarah Lucas.
CRISPR is a gene editing tool used by scientists and researchers, and also us in our genetics lab, to make specific and intentional edits to DNA.
CRISPR is made up of exactly two parts. First, we have Cas9, which serves as the molecular scissors, and then we have gRNA, which serves as a guide to tell the Cas9 exactly where to cut. Together, these two come together to make the Cas9 gRNA complex.
In the Cas9 gRNA complex, gRNA matches up and tells Cas9 exactly when to cut, as previously said. It lines up with this target site, and once that cut is made, it allows researchers to make specific edits in the DNA.
Specifically, in our genetics lab, we use the plasmid called BPK764, which contains Cas9 and an empty spot for gRNA.
To start, we did a plasmid isolation, and this is essentially preparing the backbone for later stages that will build off in the lab.
Next, we cut the plasmid with a restriction enzyme called BSA1. This isn't CRISPR. This is a restriction digest, just like we did the first couple weeks of genetics, that opens a space in order for us to insert the gRNA.
Once that space is open, gRNA, which can be customized as seen in that one assignment where we essentially customize our own DNA sequence, and that's what makes CRISPR a customizable process of gene editing.
After the gRNA was inserted, we did a ligation in order to close the nicks in the plasmid.
After the ligation, we used a mini prep in order to determine if the plasmids were purified and if the gRNA was inserted properly.
Finally, we introduced the plasmid into E. coli BL21 in order to activate the CRISPR system. BL21 allows us to make more plasmid DNA or proteins.
In summary, CRISPR, and a part of the Cas9 gRNA complex, is used to make complex gene edits. In this lab, we used a series of processes through isolation, restriction digest, ligation, as well as transformation into E. coli to study how gene editing is used within the research community.
Thanks for watching. Bye.
Bye.